and it talks about not just taxonomy alone, rather. It talks about microscopy, staining, and classification. Now, I will run through microscopy because it's something that you have done and you do also in your lab more. So let's look at this. Now, every microscope have the lenses, all right? And then we use this metric system in its calculation. When you say the metric system, you're talking about the standard of measurement, the units, okay? So meter is the major unit. Then we use decimeter, centimeter, micrometer, millimeter, micrometer, and nanometer. Let's go. Now, okay. Now, whenever you are looking at the microscope, there are four things that are very important: the wavelength of radiations. We are going to look at magnification, the resolution, and then the contrast. Now, you notice that when you go into the lab, most of the time you see a slide that is either pink, purple, sometimes you see blue. And when my students see any other color that is not pink and purple, they seem to be like, we can't relate with this image. And I keep trying to tell them that no, the issue is not about the color. Just like if I come here with different clothes today, it doesn't mean you are not going to know it's me. The issue is about the structure itself, not the color. And there are some stains that can be used to stain different slides, but that does not change the color of the slide. I mean, that does not change the content of the slide. So what we look at, and that's what all of those do. So we'll be talking about that. We stain images to give contrast. If you are taking your biology one during the time that we were still actively doing the chick cell, or for some of you, you did onion, all right? Now, you did onion and you prepare slide with it. Some of us did the chick cell, you got some cells with toothpick and put it on the slide and you look at it. But if you look at it without putting stain on it, everything will just appear white, like colorless on the slide. And you won't be able to see. They won't have contrast. Contrast is what makes you to be able to differentiate between nucleus and cytoplasm because it comes with different color. All right. So let's go. All right. I'll keep that for that. We don't need to look at that. Now, resolution. Now, resolution is what makes you to differentiate between 2 points that are close together. Now, if you have ever taken note of it on the microscope, you notice that you are looking at image and it looks blur. Now, when it looks blur, what is happening is because the resolution, you need to use a fine adjustment knob to make the resolution better. And that is if you have good resolution, it's going to show you the images clearly. All right. So resolution helps you to differentiate between two things that are on an image. Different microscope have different resolution powers, which we'll look at. Now, you see this showing you different objects and how they have been observed in different microscope with different resolutions. So if you look at this, for instance, now, the scanning turning microscope, this is the limit of what you can see. You can see between 0.01 nanometer to 10 nanometer. All right, so that is then the transmission electron microscope is able to see between 0.1 nanometer to 100 nanometers. So that tells you what you can get the best in each of them. All right, and those are those images taken from those lenses, microscope at those resolutions. Now, contrast, okay, is the difference between intensity between two objects or between an object and its background. That's what I was showing you. So what contrast does, the reason we use stain is to give it contrast. That's the major thing. One of the things we use stain for when you look at those slides on the microscope, that you can say, of course, when you're looking at those slides, you can say this is this cell, this is this cell, this is this cell. The reason you could do that is because of the contrast. And what brought the contrast? It is the staining that does that particularly. Let's go. All right. Now, we have different kinds of microscope. We have the we have the microscope that we call the bright field microscope. Okay, so we have light microscope. That's the major thing. We have light microscope. We also have electron microscope. The light microscope uses uses light as its principle, whereas the electron microscope uses electron as its principle in getting images. Please excuse me. So the light microscope uses light, while the electron microscope uses electron. Now, so the simple one, it contains one single magnifying lens. So we call it simple. There is one that is compound, which will contain multiple lenses. So you see all those lenses that you can just put in your eye, that you can just take. You see, you have some lenses that you can just hold like this and then observe something with it. That is simple because it has just one lens. But the compound have multiple lenses, which we will talk about. I'll show it to you. So it's similar to magnifying lenses, just like regular magnifying lenses. This was what Leeuwenhoek used to observe a microorganism in this experiment. Now, this is compound microscope. You see, it has a series of lenses. That's the difference. Simple have one lens, compound have multiple lenses. So because it has multiple lenses, we need to calculate the magnification eventually, which I'm going to show you in a bit. Let's go. Now, looking at this lens, this is compound lens. It's light microscope and it's compound. This is what you use in the lab. Why is this compound? This is what you use particularly in the microbiology lab. The reason it's compound is this. Look at this. It's compound. You see, you have these lenses here. You see that? You also have these lenses here. Because of that, if you have each, for instance, now, these are 4 lenses here. You have the one that we call the scanning lens. We have the low power lens, the high power lens, and the oil immersion. You want me to take it again? All right. We have the scanning lens, low power, high power, and oil immersion. The scanning have the power to magnify 4 times. The low power have the power to magnify 10 times. The one behind this here, which we call high power, have the power to magnify 40 times. The largest one, oil immersion, have the power to magnify 100 times. Now, both, whenever you look at the lens here, we call these lenses here objective lens. When you look at these objective lenses, you have to put your eye here anyway. We call this lens where you put your eye, we call it ocular lens. This is called objective lens because that is where the object will be. You know, you are going to put the object here. So that's why it's called objective lens. Now you're going to put your eyes here, and so it's called ocular eye. So how do you get the total magnification? So for instance, if you put your lens on the, currently now is on the 100x, this ocular lens magnified by 10 times itself, 10 times. It's the same lens, so they are not different. It's the same. You just have 2 for your 2 eyes. So if you want to get the total magnification for this experiment now, because the lens, the ocular lens is on, the objective lens is on 100x. So that means your total magnification will be 10 times 100. That will be 1000x. You'll notice that when you look at slides in the textbook, they always put the total magnification of the microscope they used. So if you were looking at the high power now, for instance, the high power, what would be the total magnification for this? Can you tell me? What would be the total magnification for this? 400x, because it's going to be 40 times 10. Thank you. If I use the low power, that will be worth What will be the total magnification? 100, it will be 10 times 10. And then if I use the scanning, it will be 40. That will be 4 times 10x. Right? Thank you. So that's the major thing I need to know about microscope. The labeling, of course, take note of the labeling. All right, let's go. Okay, let me talk about this a little bit. You see this here. Oh, this microscope is a little bit different from the one used in the lab, the shape, but it's the same thing. It's just like you have Toyota Camry 2010, and then you have Toyota Camry 2020. They are not different. I mean, they are Toyota Camry, but their bodies are different. So it's just like this, the one in your lab now. Of course, we still have one or two of these in our labs, but the one in the lab now is a little more recent than these ones. This is like old model, but the same thing. All right. So everything is still the same where they are located, but they just made it a little bit fancy. All right. So here now, this large one here, when you are moving it, you know that it's not moving so smooth. But you notice that this one moves so smooth, very smooth. The one that moves so smooth is called smooth or fine adjustment knob. This is the one that helps with resolution. And then the rough, this one is rough. It's called rough or coarse adjustment knob. Okay? It's either adjustment knob, focusing knob. It's the same thing. All right? This is the stage where you put a specimen and it has a clip, which we call the stage clip. All right? Okay, so let's go. I don't need to. Okay, the dark field, we really don't use it. You don't use it in your lab. But majorly, they usually use it to observe images that are quite small and doesn't have color. That's why it's called like dark field and not bright field. You know, it's just like if you want to, there are some things if you want to look at now. Let's say you have something that is shining in your room and you want to look at it. You put off the light so that you can see it. Just like, okay, very good. If I'm teaching now and I'm using PowerPoint, I'm not going to have the brightness in the room. I want to dim the light so that you can see the PowerPoint. That is that's the way the dark field works. So because of the way it works, there's a way it helps with contrast such that you can able to observe images at such point. So you see, they said, I mean, it's best for observing pale images because it can give better contrast to them. So then phase microscope, the major thing is just giving contrast to different side phase microscope. So it's used to examine living organisms or specimens that would be damaged by attaching them to slide or staining. See, the major thing is that it creates contrast very well. Just light waves out of the face. All right, are out of the face rather. There are two types of them. We have the phase contrast, and we have the differential interference contrast microscope. Let's go. We don't really do any of these. Is it have any of the? No, the microscope we actually have that we work with is this compound light microscope and also a dissecting microscope. We don't need the other ones. Yeah, we are essential. You may see, if you are not likely to see it even for facilities that have it because they are not available for what you are doing because you don't need them. All right. Now this is okay. This here is there is the fluorescence microscope and fluorescence. Think about it as light. You see something it has fluorescence, like it's light glowing on an image. So this also it's light microscope. So it uses direct ultraviolet rays on the specimen. Now the specimen will radiate the light as though it's sparking. That's what happens there. And at the long run, this image is going to have like quite a good resolution, and then it's going to have good contrast because of the light that has fluorescence. So we use it for immunofluorescence. We use it to identify pathogens and to make some protein visible. So we use it like when you want to identify markers, you want to identify some proteins, you use fluorescence. Sometimes you want to identify some antioxidants, we use the fluorescence to identify them. Right? So this is exactly what we are seeing here. So you see it beams light on the structure and then they glow. That's the major word here, they glow. That is fluorescence. Right? So that's the same thing there. You see the subject focus on will glow. Right? I really don't have to talk about this confocal. We don't need that. Okay. Now this is the same view of microscope. Just look at them. Now some microscope uses electron instead of light. So instead of beaming light on the image, it beams electron on the image. It beams an electron. Now there are two kinds of electron microscope. There's what we call the transmission and the scanning. The name tells it. The scanning is just like when you are thinking of scanning lens. All right. What the scanning does is that it scans through the image on the outside. So scanning electron lens, we call it SEM, S-E-M. It will not show you the details inside. It will show you the details on the surface. So let's say you are looking at a cell now, for instance, and you use scanning electron microscope. It will not show you the inside of the cell. It will just show you the outside. But if you use the transmission electron lens, it will show you the inside. It will pass through the whole of the image and show you the inside. It's just like if I'm walking now, I mean, if I want to see the inside of this thing now, of this phone, scanning lens will show me just the outside. It will just show me the layer of the cell if I scan it with it. But if I want to see the inside, then I have to use transmission. It will be able to show me the panel, show me the inside of the cell. That is of the image that I want to observe. That is scanning lens. I mean, that is the transmission that will do that. Right? Now, the major thing about the scanning, about the electron is this. Now, do we use the electron microscope at ACC labs when we do your microbiology or AP? No, we don't have to because there's no need for it. This microscope is able to magnify quite higher compared to what we use. Because we use lenses that magnify like 10 times, 4 times, 100 times. I mean, yeah. But these lenses are able to magnify like 10,000 times. So they are able to, we use them for detailed analysis of microscopic organisms, right? So viruses, internal cellular structures, those are what we use them for. We don't use them for the basic, you want to look at slides that can be observed with 40x. Most of our images with 40x, you are good. With the high power, you are good. So those are electron microscopes. That's it there. Right? Those are electron microscopes there. So you can see this here now, you can see it showing you the image here. And this is transmission. Of course, you can see the inside. You're not just seeing the outside. Scanning will show you the outside, but you see transmission showing the details inside the cell because it will penetrate its electron into the cell. Right? Right? This here now is scanning. You can see, you can only see the surface. It's showing you the surface. This is scanning. You are seeing a detailed analysis of the surface. You can see this here is scanning. You are seeing the detailed analysis of the surface. Here is showing you the 3D of the surface. But look at what you saw in transmission. You can see the details, not just the surface alone. Exactly. That's exactly the concept. Thank you. Of course, that's it. All right. That's exactly what we are talking about. What do we call this thing? There's something I'm trying to remember. X-ray uses radiations, right? So, and that's about the way it works too. Just to give you a picture of it. Now, this is scanning electron microscope, showing you image. Now, I don't really need to talk about it. Let's go to talk about the things. I want to be able to talk about the things that I know you need. Okay, you have talked about it, just differentiating, making a table of the things we've discussed. Now, staining. Staining. I already told you why we stain. Number one is you won't be able to have, if when you did the experiment, for some of you that did it, and if you didn't do it, it's okay. But if you bring out your cheek cells now, they are colorless. If you bring out your cells, the cells, they are colorless. If you put them on a slide, the slide itself is colorless and transparent. So you're not going to see anything on it. You may see that there's something there, but you just don't see lines. Nothing. So the purpose of staining is so that we can stain and identify the different features. Now, you see, when you are looking at even tissues on the microscope, you can say this is the nucleus. You can say, oh, the dark spots are the nucleus. You can say, oh, the nucleolus is there. It's because they have been stained. If they were colorless, you will not be able to see them. So the purpose of stain helps to give you resolution. It helps to give contrast so that you can see your image to details. All right. Now, if not, you will not see the image. You will not see the resolution will be bad. The contrast will not be okay. Now, so the purpose of staining is so that you can observe it on the microscope. All right. Of course, you have to be careful not to overstain your specimen based on the protocol. But then the purpose of staining is to be able to observe your specimen. Right. So this is it. Whenever you want to make a slide, you put your specimen on the slide, then you air dry it. Of course, you air dry. Most of you will be doing that in the lab at the moment already. You air dry. If you don't air dry, the specimen can wash away, because by the time you put your stain is liquid. They can just wash it away. If you're not careful, the time when you are washing, if you don't air dry, the specimen can easily wash away. But if you air dry, it stays there. Another thing to do is sometimes you just pass through the flame. You don't let it stay on the flame because sometimes you depending on how you want to do, but you don't want to put too much of that. Now, when you heat fix, we call this heat fixing. When you heat fixing does two things. Number one, it makes the specimen to stay on the slide. Number two, it also kills the bacteria. And sometimes that's what you want to do. So heat fix does that sometimes as well. That's part of the purpose sometimes. Now, principles of staining. Now, dyes are used as stain, and they are usually salt. Chromophore is a kind of stain that is colored, a colored portion of that. Now, take note, please. Any dye that is acidic will not stain acidic part of the cell. It will stain the basic side of the cell. So any side that is acidic will stain basic part of the cell. Any part that is any stain that is basic will stain acidic part of the cell. So acidic dye stains alkaline. Alkaline is the same thing as basic. Please don't confuse that. Alkaline is the same thing as basic. So acidic stain or acidic dye will stain basic structure. And basic stain will stain acidic structure. Please excuse me. Give me just a moment. I will be back. So acid stain stains basic part of the cell, and then basic stain the acidic part of the cell. So when you see different part of the stain of the cell stained, is because of cell stain, is because there are different stains that may have been used, and then the ways they pick. Now, when you normally see that, like for instance, now the nucleus is acidic. So basic stain will stain the nucleus. Acidic stain will not stain the nucleus. All right. So when you're looking at stain like H and E, imatoxylin and eosin stain, so different part of the stain picks the different components of the slide. I mean, of the specimen. Now, simple stain. When you say simple stain, it's just one stain. Just one stain. It can be like crystal violet. You use it to stain something. You are just using that one stain to stain the object. You can use safranin. It's just one. Methylene blue. It's just one. Like now, particularly when you are doing your cheek cell. If you still remember, you will remember that you use methylene blue. When you do onion in the lab, you use methylene blue. Those stains are just single stain. So anytime you are doing a staining techniques and you are just using one stain, you are not using two different kind of stain for differential or selective. That is simple stain. All right. Now, this kind of stain is simple stain. You can see it's just picking up one stain here. Generally, those are simple stain. Now, all right. So like this now, it's showing you different kind of cells, cell shape. You see, that's showing you different kind of cell shapes right there. Right. Those are simple stain. Now, this should not be a problem because when you look at it, depends on the kind of stain that it is, and then what is reacting with the components of the cell that is staining, right? So that determines the color that you're going to see. Okay, now differential stain, however, all these stains here, they are differential stain. They use more than one dye, for instance, and we call it differential because it's going to differentiate something for you. It's going to differentiate the nucleus and the cytoplasm. It's going to differentiate some structures like that. So that's why it's called differential. It will be used to differentiate different parts of the cell. For instance, now, if you are staining for histological stains now, you use H and E. We call it H and E, imatoxylin and eosin. The imatoxylin, for instance, it is a basic dye. So since it's a basic dye, it's going to stain the nucleus. Does that make sense? So that will stain the nucleus. And so usually you always see that that area will be blue, purple, as the case may be. All right. So usually when you get to that, you see those slides, you see the purple on the nucleus is because imatoxylin is the one that stained it. All right. Eosin, which is like somehow pink, stains the cytoplasm. So you see the majority of those slides are pink because eosin stain pick them up. Eosin is acidic, so it's going to stain the cytoplasm. So that's the way it's differential slide stain. For instance, now hematoxylin and eosin, once you are done staining, you can tell that this is the nucleus on that slide and this is the cytoplasm. So it has differentiated them. And it's usually two stains. So you use one first, you rinse, and then you use another. You use one, you rinse, you use another. All right? That is hematoxylin or any other kind of differential stain. Now, gram stain is also differential stain. Gram stain is also differential stain. Now, when you are using gram stain, you are going to use crystal violet, and then you are going to use safranin. Now, because of that, I'm going to show you, I think I should have the images that shows that, but you're going to see, have you ever heard of gram-positive and gram-negative bacteria? Okay, now.
Yes, I know about gram-positive, gram-negative.
Very good. So gram-positive bacteria, they are going to be, you see them, they will be purple. And then gram-negative, you see them, they will be red. They will be pinkish. Now, thank you. Now, when you see them, is because the stains that they picked up are different. I'll show you an image now. Now, the reason they do that is because the gram-positive bacteria, they have what we call peptidoglycan. Both of them have peptidoglycan, but the peptidoglycan is what dominates the cell wall of gram-positive bacteria. So they have about 90% of their cell wall is peptidoglycan. And because of that, they pick up that gram stain. Whereas the ones that are gram negative will not pick it up. And so they will not pick up the stain. And because their own peptidoglycan is less than 10%, it's limited. It's just like you're saying something is here, but you really can't trace it. That's the way gram negative are. They have the peptidoglycan, but it's quite small and negligible for the cells to pick up. All right. So I said those stain there, for instance, is crystal violet and safranin. The safranin is going to the crystal violet is what the gram-positive will pick up, which is why they will turn purple, and then the safranin is the one that the gram-negative picks up. All right, so at this now, for instance, this is a gram stain, and its job is to differentiate, to show you which one is gram-positive and which one is gram-negative. So you see, first thing, you stain with crystal violet, which is like purple. Now, when you are done staining, what's going to happen? All the cells will be stained purple. But all the cells are actually stained purple, but not all of them have picked up purple. There's different. Not all of them picked up the stain. The fact that you put a stain on a surface and then it all made the surface purple does not mean the whole cells reacted with the stain. Now, what's going to happen? The next thing, by the time you rinse the slide, the one that didn't retain it, but it just covered their surface, it will go off. But the one that retained it will stay there even if you wash it. But one thing you have to do first before you wash it, we use iodine on it. So after you stain with crystal violet, then you're going to put iodine. Now, what will iodine do? We call iodine a mordant. It's like a sealant. It's like you want to seal up the stain that crystal violet has stained for the specimen that has picked it up, actually, so that you don't wash it off from them. But it will not still affect the ones that didn't pick it up, that is just on their surface. So the first, all the cells are stained. Second, you're going to use a mordant, all right, so that it can seal up the ones that actually took it, that actually attracted the stain. The next thing, you're going to wash it now with ethanol. Ethanol, what it is going to do is that it's going to wash up all the excess purple that are there, all the excess crystal valence. What do we mean by excess? The ones that are not really absorbed by the gram-positive cells. All right. So next thing is this. You notice it has washed up completely the excess one here. Then the next thing is now you go ahead and stain with safranin, which is expected to stain the gram-negative bacteria. Now you see all the cells that were colorless here because of the washing with alcohol, we call it decolorization. All the cells that were decolorized has now become pink because safranin is pink, is that they've picked up safranin. So that kind of, I know I've taken time on gram staining. That is a differential staining. What does this kind of staining does? It has helped us to differentiate now between the gram-positive bacteria, which are the cells that have peptidoglycan dominating their cell wall, and the cells that have no peptidoglycan, lower peptidoglycan dominating their cell wall. Of course, it's okay if you don't know yet. Gram-negative bacteria are more resilient. It's difficult to kill them compared to gram-positive bacteria because of the nature of their cell wall. All right, now this is acid fast. I don't need to talk too much about it again. Let's go. All right, they are also all differential. This is endospore. Some pick up the spin, the endospore. I think I talked about endospore the other day. All right, so Those are different. All of those are differential staining where they are used to differentiate different kinds of specimen or part of the specimen. All right. Okay. So I've talked about, this is what I was talking about earlier, hematoxylin and eosin stain. Right. Now, there are some special stains. We call them negative stain is a special stain. Flagellar stain is a special stain. Fluorescent stain is a special stain. Now, the major thing we use the special stain for is to identify microbial structures. That's the major thing it's used for. So they are not, usually they are not the stain that we use in the lab majorly. You really don't use it. We don't, except for like major experiment. Like now I've used this fluorescent before for an experiment that has to do with pancreatic cancer. All right, let's go. So this here is negative stain. It's used to stain the capsule. So you can see the capsule. The capsule will be this is K pneumonia. Klebsiella pneumonia is a kind of bacteria that causes pneumonia, right? And let me see if there's anything else I want to point to. Right? Of course, you can see that's the bacteria itself, and then the capsule is the clear area that you are seeing all around it. Right? This flagellar stain, the major thing it's going to do is to identify the flagella. That's the major thing there. So you see the flagella all around. And all right, this highlight the different kind of stains that you have looked at. Right, let me see if there's, okay, good. I'll talk about taxonomy, which is the last part I will be talking about. Now, taxonomy, what does it mean? Another word for taxonomy is simply grouping. Actually, taxon means group. So taxonomy is another word for grouping. Now, why do we do taxonomy? Is to make study of biology easy, to make classification of organisms easy, and it helps to categorize organisms particularly. Now, so if you discover a bacteria now, you already know what the characteristics of the bacteria will be. You know that it's prokaryotic cell, that's number one. You know that it has circular DNA, number two. You know it's going to be unicellular, number three. How do you know all of that? Because we already know that prokaryotic organisms have those features. So taxonomy talks about classification of organisms, naming them, all right, and identify them. Right. It also helps to organize large amount of information about organisms. It makes predictions based on knowledge of similar organisms, like what I just said. And it's going to help evolutionary connection to connect organisms based on how they evolve. All right. That helps to study evolution. Of course, many times, if we don't have a track of the existence of organisms, you're not even going to know when an organism evolved anyway. Right. Okay. Now, the guy that does the classification that is called Carolius Linnaeus. So usually we call it linear system of classification based on his name, Linnaeus. So we call it linear system of classification. We call it binomial classification because he used two names, just like first name and last name, species and genus. That's what he used in naming them. So a system classifies organisms based on common characteristics. He grouped them according to how they can interbreed, which is species. So species are placed in a group, organisms that can interbreed together and produce live viable organism is what we call species. Like, is it horse and donkey now giving rise to mule? That is not, because they are not of the same species, they are going to give rise to organisms that cannot reproduce. All right. So that is what we call, so a species means that if they mate, both of them must be able to give rise to organism, not just give rise to organism, they must be able to give rise to viable organism. So horse and donkey mating and producing a mule cannot reproduce. So that makes that proves that they are not, even though they can reproduce together, but the fact that their offspring cannot reproduce, it shows that they don't belong to the same species. Right? So they use two systems of naming. So we call it binomial nomenclature. Nomenclature simply means name. Binomial means two names. All right? So look at these now. This is taxonomy, classification. All right? The domain, the largest domain is the largest classification of organisms. So we have bacteria, archaea, and eukarya. That's the largest. And of course, I told you bacteria and archaea, they are both prokaryotic cells, and eukarya are eukaryotic cells. Excuse me.